Hi guys, welcome back. All right, so we continue our lecture and to talk about the membrane potential. So membrane potential basically is about that. Let me show you what is membrane potential. So when we in in um in A and B or anatomy physiology. One thing you need to keep in mind is always like when you learn about the terminology, a new terminology, you always think about two things, location and the function. Where is it and what does it do? So where is it? Membrane potential is a membrane. So it's right here. So here is the membrane. Cytosol means that inside of cell, and this is outside of cell. Plasma membrane, we mentioned about that. This is the cell membrane. The barrier surrounding the cell. And so you can see that if you put the electrode inside outside, you see that it's always inside is always negative. Outside is like shared space between cells. So we consider this as the ground, like it's the common ground of all other cells, all cells. And for each cell, we measure what is inside. And so we can measure it as negative. And you will see this throughout the every cell in the human body, that inside of the cell, the charge is negative, somewhere like minus 50 or 60 millivolt MV. Yeah, so that is what we got. So this is membrane potential. So location is on the membrane. Function is a phenomena. It's just and it will become quite important. This is the foundation for electrophysiology. Any like electro thing in the humans in the organism is based on this. So this is important. So that's memory. We are going to talk about that. Before we talk about that, I want to review one thing that we just mentioned earlier, which is the active transport. So that's this thing here. This thing here. So sodium potassium pump. Every cell, as I mentioned before, every cell has this pump. So every cell will have this pump to do one thing, pumping sodium and pumping potassium. In addition, not in addition, so that's that. All right. So it pumping sodium, pumping potassium. We need to know that it pumps 3 out, pump 2 in, pump 3 sodiums out, pump 2 potassiums in. These pumps will result in two consequences. One is that you're gonna accumulate sodiums outside of cell, and you're gonna accumulate potassium inside of cell. So let me draw it again right here. Let me use my tool, pencil, to draw it again right here. I already have my pencil. So this is the cell. So with this pump, it will lead to one consequence. The first consequence that you're gonna accumulate potassium inside of cell. Potassium inside of cell. Sodium outside, it's gonna flow away. So it's out, it's gone. But you're gonna accumulate potassium inside of cell. Okay, you can put sodium outside, that's fine. Sodium outside. Everything is outside. Everything else is outside. But potassium is inside. Yeah. So you trap potassium inside of the cell. So that's first thing. You trap potassium inside of cell. Second thing is that you can see that sodium and the potassium are positive charge ion. So these are positive charge. These are positive charge, positively charged. So you have more positive charge to push out, and you only push in less positively charged ion. So you have 3 out, 2 in, 3 out, 2 in. So gradually you have 3 out, 2 in, 3 out, 2 in, 3 out, 2 in. Gradually you build that diversity, you build that polarity difference. And so inside is not so much, but you have more positive outside. So you create that polarity. Inside is not so much, but you have more positive outside. So that's that. So that's the second consequence because these two numbers are not the same. 3 out, 2 in. Yeah, so that's that. This gave the foundation that the membrane is not equal. The charge across the membranes are not equal. Inside is relatively more negative, and outside is relatively more positive. That is the sign of a living cell. A living cell has the ATP. A living cell gonna pump. A living cell gonna create a negative membrane potential. So that's that. If you don't see that charge difference, if you don't see that polarity, this cell is a dead cell. Yeah. So that's the foundation. Right, so that's that. Membrane potential is driven by sodium potassium pump. Oh, I forgot to mention sodium potassium pump is also called sodium potassium ATPase. I mentioned it here. Sodium potassium pump is also called sodium potassium ATPase because it requires ATP. ACE means like lysis. It break apart the ATP, utilize it to do its work. So that's that. So this one basically is single-handed create membrane potential. So first thing is that we can see that the ion distribution between intracellular space versus extracellular space. You can see a lot of things here, different kinds of the ion, different kind of ions. But the only thing you need to memorize is that potassium is high inside of cell. Right here, potassium. See potassium very low. Potassium is high inside of cell. Everything else are outside. So that's very simple. Everything else are outside. Inside of cell, you have potassium because of this sodium potassium pump. Second thing is because the number is different. 2K in, 3 Na plus out. The number is different. So you create that negative, relatively negative charge inside of cell relative to the extracellular space. So that's what we got here. Every cell, every human cell has a sodium potassium pump, as sodium potassium pumps. The consequence is that it gonna lead to a negative charged membrane potential in the cell. So every cell, with the skin, whether that's nerve, whether that's blood cells, you're gonna see the membrane potential to be negative. So that's that. Very simple, right? Membrane potential. The next thing that we are going to learn is that, all right, so every cell has a negative membrane potential, but some cells have more than that, has more than that. Some other cells will utilize this to do different things. We call this group of cells excitable cells. Is that now the membrane potential is negative, right? You have more positive outside, relatively more negative inside. Some other cell will use it to do some manipulation and the big and to perform its function. We call these cell are excitable cell. They can be excited. Excitable cells, there are many, but generally speaking, nerve cells and the muscle cells are excitable cells. Yeah, they can get excited. When they get excited, they will change the membrane potential. They will naturalize it. They will depolarize it. So this is polarized, right? They will depolarize, make it neutral, make it equal. And then that will show a signal to surrounding things. And then they will return back to negative again. So they can depolarize it and then repolarize it. And this action is called action potential. Yeah, so excitable cell will change it to create an action potential. So let's take a look. Every cell has a sodium-potassium pump, but excitable cells has two more things. They have, in addition to the sodium-potassium pump, they also have voltage-gated sodium channel and the voltage-gated potassium channel. So that's two other things. Sodium-potassium pump lay the foundation of a negative membrane potential. Voltage-gated sodium channel and voltage-gated potassium channel allow these cells to generate action potential. Yeah, so let's take a look. So this is a figure of this action potential. So you can see that this is time. So cells, times goes on. Cell is not excited. The membrane potential will be negative, say minus 70 millivolt. So it's negative, right? Time goes on, it's always negative. Now, when you excite it, if this can be excitable, then it will generate action potential. So you can see that its membrane potential will change from negative to be 0. It sometimes overshoot a little bit. Depolarize. It was polarized. Now it depolarized. So this phase is called depolarization. And then very quickly, it will return back to polarization. So we call it repolarization. So we have the depolarization. When you excite it above a threshold, you need to excite it. You need to stimulate it strong enough to move above the threshold. Then it will trigger its depolarization. Everything like it was polarized become neutral and sometimes overshoot a little bit. And then repolarization. So it's excited, come back and coming out and back. Coming out and back. Yeah. So that's action potential. This one is driven by these two membrane protein. You got to have a voltage gated sodium channel. It's gated. It allows sodium to go through. It's gated by the voltage. So this voltage is the charge on the membrane. You also have another one, voltage gated potassium channel. So this channel allows potassium to go through and it's gated by the membrane charge. So that's that. And the action potential, you basically has depolarization, two major phase. You have a depolarization phase, and then you have the repolarization phase. So first is that you need to stimulate these cells above a threshold. So stimulation is needed to initiate this action potential, and this stimulation has to be above the threshold. Once it's above the threshold, then you trigger it, it will create a depolarization, very short period of time, 1 millisecond, followed by very quick return back to the baseline repolarization. So depolarization is driven by here, threshold. So you can stimulate it, you can poke it, but if it's not above threshold, it's just coming out and back, coming out back. So it doesn't generate the depolarization. It doesn't really depolarize it. Only until your stimulus is above the threshold, then you trigger its depolarization and the repolarization. So just like your touch, right? You stimulate it, and it has to be strong enough, so it trigger action potential. Then you can transmit that signal into your brain, so you can receive that touching sensation, right? Then you have the depolarization. Depolarization is driven by voltage gated sodium channel. So basically that this channel right here. So here is the sodium channel. All right. So you have the voltage gated sodium channel. You have voltage gated potassium channel. When your membrane is stimulus, the charge change, it increased a little bit, but it's not too much. However, that increase, if it's above the threshold, your voltage gated sodium channel gonna pick it up. And when it pick it up, this channel will open. When this channel open, it will allow sodium to go through. All right, so let's go kind of like review of what we have learned. This is a facilitated diffusion. A channel, sodium. Sodium will go through by diffusion. Diffusion is a movement of the molecule from high concentration area to low concentration area. Now, when this channel open, you have a cell. You have this channel. When this channel open, it allows sodiums to go through. All right, so sodium can freely diffuse across now. Sodium can go through. Yes. So, and this movement is moving from high concentration to low concentration. So you kind of have to decide, right? You can kind of like ask yourself, will sodium tend to move in more or move out more? And the question is, basically, where do we have higher concentration of sodium? Is that outside or is that inside? And the answer is outside, right? So we know that out inside you only have potassium. So sodium is more are outside. So you have a lot of sodiums outside. And so when this channel opens, sodium will flush in, like coming in, right? Sodium will come in because they're gonna move from high concentration to low concentration. And with this sodium coming in, sodium bring a positive charge ion, they're gonna make inside to be very quickly to be positive. So that depolarize it, remove that negative environment to become positive again. Yeah, so that's that. Depolarization is driven by voltage gated sodium channel. Sodium will influx from outside to inside of a cell. And then this channel close. Very quickly, when the channel, when the membrane potential reach to like 0 or a little bit positive, then potassium channel open. And so right here. So when potassium channel open, again, right here. So a cell, you have the voltage gated potassium channel right here. So again, here you have the voltage gated potassium channels. This is again a facilitated diffusion. So the question is, it allows potassium to go through. So this allows potassium to go through. And then we know that molecule move from high concentration to low concentration. So the question is, where do we have high concentration of potassium? And then we know that inside of cell, you have high concentration of potassium right here. You have a lot of potassium inside of cell. So when this channel open, a lot of potassium will go out, right? So it will diffuse out. It will diffuse out. It will diffuse out. And when it diffuse out, this is a positive charge ion. It will make inside to be negative again. And so it causes the membrane potential to become negative. It causes inside to be negative. It repolarize. It make it polarize again. And so this is the repolarization phase. It's driven by voltage-gated potassium channel. And with this channel open, potassium will move from intracellular space to extracellular space. So like you see here, this voltage-gated channel, it opens, potassium goes out. This is a concentration gradient from high concentration, which is inside, to low concentration, which is outside. So that basically summarize everything you need to know about the action potential. So this one summarize it before any excitement, any stimulus, cell maintain a negative memory potential. You have negative charge inside of cell, positive charge. Here it shows neuron. Neuron is an example of the excitable cell. And so you can see it's a negative inside, positive outside. You have more potassium potassium ion in the cell, and more sodium ion out of cell. When you stimulate it, the first thing you see is a sodium channel, sodium right here, sodium channel open, sodium channel open. When sodium channel open, voltage gated sodium channel open right here, voltage gated sodium channel open. This one is, first of all, the resting memory potential is driven by sodium potassium ATPase. So you have this pump causing high concentration potassium intracellularly, and also memory potential to be negative. When you stimulate it, you change the charge, you make it a little bit, like it's not a lot, but you stimulate it, and that can be picking up by voltage gated sodium channel. And this channel will open, then it allows sodium to go through freely, and from high concentration to low concentration. So sodium will go in, and then it will bring the positive charge into the cell, and so it caused depolarization. So it was negative, and then here positive, right? Positive coming in, negative and positive coming in, and then it closed. Then you have the repolarization. Basically, it's driven by the voltage gated potassium channel. You will see that it will coming back because this channel open, potassium freely diffuse across from high concentration to low concentration. While they move out, they bring this positive charge out of the cell to make the inside to be negative again. And so it's means regain that polarization. And then it goes. And then you have this sodium potassium pump to maintain or to create the resting memory potential, which is negative throughout our body. Yeah. So that I think that summarizes everything we need to know. I think this is a last picture, maybe not last, but here is a summary again. So this is the action potential. Before you stimulate it, it remain as a resting memory potential, which is always negative. Question is, what caused it to be negative? And at this point, you should know that the things to cause a memory potential to be negative is because sodium potassium pump, and every cell has it. Now, when you stimulate it, if it's weak stimulus, it won't cause any change. It won't stimulate an action potential. But if it's strong enough to push it above the threshold, then you're gonna trigger a voltage-gated sodium channel. Sodium go in, causing depolarization. Very quickly, it close, followed by voltage-gated potassium channel open, allow potassium to go out, and then driven the repolarization. And then when it goes out, sometimes it run too fast, make it too negative. And then it's called hyper, too much. Polarization, hyper means too much. Polarization means it's polarized. But you have this sodium potassium pump to gradually regain them into the resting memory potential. We see this action potential in the excitable cells, and two major type of cells that are excitable are nerve cells and the muscle cells. So I think in my last slides, I'm gonna show you the application or this scenario in a nerve cell. So here I show you the nerve cell. A nerve cell in our nervous system lecture, we kind of go through what a nerve cell look like. A nerve cell basically has a cell body, it's called soma, and an axon. So this membrane is negative, has a negative membrane potential. But if it receive the stimulus, like your touch, like you see something or you smell something, acting on it, you're gonna trigger the action potential. So it will get depolarized. So you can see that in the beginning is negative, inside is resting membrane potential. But when you stimulate, you first trigger the voltage-gated sodium channel. So you cause it depolarization. With this depolarization, even though it's just locally allow sodium to go in. Let me draw it here. Allow sodium to go in. Even though it's very locally, allow sodium to go in. This sodium gonna diffuse across. And when it diffuse across, it gonna trigger an action potential. So this action potential gonna propagate from the soma, from the location where they receive the stimulus, all the way into the axonal terminal. So you can see that depolarization, repolarization, this gonna propagate from the beginning of the axon toward the end of the axon. Yeah. So we call this nerve impulse. You need to know nerve axon is very long. For example, when you touch something, this nerve has the axon go all the way into your spinal cord. So you definitely can use your electrode to detect this impulse from your finger and to see its transportation to your spinal cord. So that this nerve impulse is basically to describe the action potential traveling along the axon, basically. Yeah, along a neuron, but basically it's along the axon. So that's that. And then so you can deliver that signal. Say this is the end is let's say fragment, right? So you put perfume on, fragment, binds on it, and then it trigger action potential. This one will go into your brain, so you can smell it. So that's how you deliver that signal. On one end, you receive that stimulus. In the axonal terminal, it will tell it into your brain because that action potential gonna travel down there. Yeah, and this transportation is very fast. Yeah. So that ends our lecture, and hope it helps you to understand the importance of the cell membrane, and also from there to understand the membrane potential and the action potential. Let me know if you have any questions. I will see you next time. Bye bye.